Sudip was joined my lab in fall 2018. So he got his BS and MS degree from Dhaka University in biochemistry and molecular biology. We are happy to have him in the lab, and he's involved in several projects. And today he will share one of his project about starch branching enzyme. Yeah, please go ahead, Sudip.
Yeah, can you hear me and see my slide?
We can hear it and see it.
Okay. Good afternoon, everyone. Thanks, Dr. Septi, for introducing me. I mainly work on rice and peanut genome editing, but today I'm going to present one of my PhD thesis work, which is increasing level of resistant starch in rice using CRISPR Cas9 multiplex genome editing. So rice is a staple food for more than half of total global population. According to the USDA 2019 report, the USA is a major rice exporter, which valued at $1.9 billion. Increasing yields as well as improving grain quality and nutrition for a healthy rice diet would also be essential. Moreover, nowadays, different diet-related chronic diseases like diabetes, hypertension, and obesity are very common in both developed and developing countries. As we all know, rice is an excellent source of starch, and starch is composed of amylopectin and amylose. There are some difference between these two components. Amylopectin is highly branched polysaccharide with higher degree of polymerization compared to amylose. One of the major characteristics in cooked foods or rice, amylose molecule rapidly precipitate and form complexes which are difficult to digestion. On the other hand, the amylopectin molecule form complexes slowly due to its highly branched structure and degree of polymerization and more easily digestible. So amylose containing food product ultimately control the blood glucose level because they have lower glycemic index. On the other hand, the amylopectin increase the blood glucose level due to higher glycemic index. Therefore, high amylose product means the high resistance starch content, which are less easily digestible and absorbed in the stomach or small intestine and directly pass to the large intestine, which helps to control our blood glucose level. Let's see the starch biosynthesis pathway in rice. There are 4 enzymes responsible for starch biosynthesis, which are pyrophosphorylase, starch synthase, starch branching enzyme, and starch debranching enzymes. Granule-bound starch synthase 1, which is insoluble starch synthase, enzyme control the synthesis of amylose in rice, while the soluble starch synthase, starch branching enzyme, and starch debranching enzyme together control the synthesis of amylopectin. It has been already reported amylose content or resistant starch content can be increased by 2 ways, either through overexpression of a suitable granule-bound starch synthase allele or suppressing the expression of starch branching enzymes involved in amylopectin biosynthesis. In this study, we only focused on 4 starch branching enzymes. So our objectives of this study to increase the resistant starch or amylose content in rice by knocking out 4 starch branching genes through the genome editing. There are some advantages genome editing over the other approaches. With the genome editing, it takes less time compared to transgenic and conventional breeding. And it also less genetic effect because we normally targeted specific region or genes. On the other hand, for conventional breeding, one of the major advantage, there is no biosafety regulation is required, but transgenic approach, it should be maintained strictly biosafety regulation. But for genome editing, biosafety regulation sometimes vary from country to country and different process also. Like European countries need more, they normally strictly maintain the biosafety regulation for processing the genome editing crops. So there are some components needed for genome editing. First, we need target DNA sequence with PAM site, and Cas9, which is an endonuclease, cuts the target DNA sequence, and the guide RNA, which normally guides the Cas9 for binding to the specific target DNA sequence. When these complexes form, the Cas9 cuts the target DNA, and DNA is repaired by two mechanisms, which are non-homologous end joining and homology-directed repair. In case of non-homologous end joining, gene disruption are occurred by small insertion or deletion. On the other hand, for homology-directed repair, donor tablet need to be given, and gene correction or gene insertion occurred by homology recombination process. There is a great news in this year. Two scientists rewarded the Nobel Prize in Chemistry for their development of a method for genome editing. This is also fascinating news for us who works in the genome editing topics. So in this study, we use Agrobacterium mediated transformation. For this, first we need to transfer a vector which contains Cas9 and the guide RNA inside the Agrobacterium. And when we infect the plant cell with the Agrobacterium, the tDNA containing Cas9 and the guide RNA inserted into plant genome. Then Cas9 and the guide RNA are synthesized from this tDNA. And finally, Cas9 cuts the genomic DNA with the help of guide RNA. And DNA is repaired by non-homologous end joining or homology-directed repair mechanism, which I have described in the previous slide. So this is the overall workflow in this study. We first identify the sequence of 4 starch branching enzyme genes and design the guide RNA in this target region. And then we check the efficiency of the guide RNA by in vitro digestion. Then we developed CRISPR-Cas9 construct, followed by Agrobacterium-mediated transformation in rice. And finally, we detect the positive transformant by PCR and mutation analysis of this transformant through the Sanger sequencing and Amplicon-based Miseq analysis. For designing the guide RNA, we used two web-based tools, which is CRISPR-P and CRISPR Direct. After designing guide RNA for all 4 starch branching genes, we ordered them commercially. This schematic diagram will show how in vitro digestion works. Suppose we have a 1.6 kilobase per PCR product, and this is a guide RNA position. After adding Cas9 and the guide RNA, this PCR product are chopped down into 2 fragments. So this strategy we were used for checking our guide RNA efficiency. So here you can see the result. So this is the undigested PCR product for our target starch branching enzyme genes. And after adding guide RNA and the Cas9, we found the fragmented product. So we can infer here that all the guide RNA we have designed efficiently cut all SBE gene targets. Then we cloned our designed guide RNA into a vector. For this, we used endogenous tRNA processing system. One of the advantages of this tRNA processing system is only a single promoter can overexpress multiple guide RNA at a time, because in our experiment, we use 4 types of guide RNA. Then we designed tRNA guide RNA construct and clone into a vector, which is PRGB32 or destination vector, because it is more or less rice-specific transformation. So after cloning, we confirm by restriction digestion and the sequencing. In this gel photo, you can see that the positive clones have different size compared to blank plasmid. Then we transfer the positive vector into Agrobacterium by heat shock transformation method. We did Agrobacterium transformation in rice. For this work, Marco Mayra Molina from Multicrop Transformation Facility helped us. So for the rice variety, we use Presidio. There are specific reasons for this, because it is a semi-dwarf rice variety and it is Texas variety. It is high yielding as well as high grain quality, and one of the most planted in-bit rice cultivar in Texas. So this is the different growth stages of transform plant. After regeneration from the tissue culture, we transferred the seedling to MS media for some days. Then we transferred to hydroponic solution for adaptation to the outer environment, and finally transferred to soil. And we keep the transform plant inside the growth chamber according to follow the proper biosafety protocol. This is the tDNA border of our desired construct. As you know, during the Agrobacterium transformation, normally tDNA inserted into the plant genome. So to check the presence of the transgene, we design 2 sets of primer. One set covers tRNA guide RNA region and another one Cas9 region. And we did the PCR with these 2 sets of primer to check the presence of the transgene in our tissue culture generator plant. So in these 2 gel photos, you can see there are the transform plant and the positive control gave positive and desired band size. On the other hand, negative control and the wild type did not give any band. So altogether, we have checked 38 plants which generated from the tissue culture, and we found all our plants is transgene positives. So we can say here that the transformation efficiency is 100% for our experiment. Then we check the mutation by Sanger sequencing and also Amplicon-based sequencing through Miseq. For Sanger sequencing, first we amplified the region of target and cloned into a vector and sent the plasmid for Sanger sequencing, and we did data analysis in BenchLink platform. And for Sanger Amplicon-based sequencing, there are some advantages over the Sanger because it is cost-effective, time-efficient. We can identify and distinguish the mutation pattern more easily, and we can run more samples at a time. Here I will show you how we prepared the Amplicon sample for Miseq analysis. First, we amplified target region by PCR. Here, the primer set overhang with the second PCR, which is called indexing PCR. So every step of PCR, we clean up the sample with magnetic beads. And this is our final library for Amplicon sequencing. And then we quantify our PCR product by qubit 4 and check the quality by Agilent step station at TIX lab. After checking the quality, we send the library at TIX lab for Miseq sequencing analysis. And they gave us sequencing read at FASTA format. And finally, we did the data analysis through the Python script, which is CRISPR. Before going to show my mutation result, I want to introduce what type of mutation pattern we got. As you know, rice is a diploid species, and every gene has normally 2 alleles. In the wild type plant, in the both allele, there is no mutation. But when there is only a single mutation for one allele, it is called monoallelic mutation. But when the same type of mutation is present for both allele, this is called biallelic homozygous mutation. And when different mutation for the both allele, it is called biallelic heterozygous mutation. So we did analysis the mutation for all our 38 tissue culture generated plant, but here I will show the mutation result only for transform plant, and later I will show the overall picture of our 38 plants. So this is our first target, SB1 target gene. The wild type plants here, both allele shows similar sequences, and green letter represents the guide RNA region. And all 4 plants gave same type of deletion. One allele gave 23 base pair deletion, and another allele 3 base pair deletion. So due to the different types of mutation, it is biallelic heterozygous mutation. So all our 4 plants, we found all the plants are biallelic heterozygous mutation. For our second target, we found here deletion, substitution, and also insertion. So overall result is 2 plants showed biallelic heterozygous, 1 plant gave biallelic homozygous, and 1 plant monoallelic. For gene target 3, we found one base pair deletion, and one of the plant, which is plant 3, transform plant, and that gave large deletion, 59 base pair deletion. And overall result is 2 plants showed biallelic homozygous and 2 plants monoallelic. This is our last target, SB4. And here we found substitution and the deletion. And overall result is 2 plants gave biallelic heterozygous and 2 plants biallelic homozygous. So I already told we have found 30 in total, 38 transform plant, and we have checked all of the plants, what type of mutation is there. And we found different combination of gene mutation here. So here we found 10 plants where all of the 4 genes are mutated. And unfortunately, we also get 8 plants where we did not find any type of mutation. So in this result, we can infer that overall mutation efficiency is 78%. 78% out of 38, we found 30 plants which plants at least 1 gene mutation. So then we check the frequency of the mutation in respect to biallelic heterozygous monoallelic and biallelic homozygous through the overall plants and the gene target. For SB1 and SB4 target, we found higher percentage of biallelic heterozygous mutation. And for SB3 and SB2 gene target, we got higher percentage of monoallelic mutation. So we also check the frequency of mutation in respective to deletion, insertion, and substitution. Interestingly, all our gene target gave higher percentage of deletion mutation compared to insertion and substitution. So it is very common for genome editing and CRISPR-Cas9 genome editing in plants result. Then we check the expression analysis of all 4 genes in both wild type and our transform plant. For this, we isolate the RNA from leaves of wild type and for starch branching enzyme mutated plants. We amplified the cDNA by reverse transcriptase PCR and expression analysis by semi-quantitative RT-PCR. First, we normalize our samples by housekeeping gene, which is elongation factor alpha for rice. Then we did expression analysis all 4 genes. Interestingly here, the gene target SB1 gave multiple bands. And when we see the mutation pattern, it shows it gave biallelic heterozygous mutation. So that's why it gave multiple band during the expression result. But if you see the comparison between wild type and the transform plant, we did not find that much variation from wild type and the transform plant expression. So here, due to the mutation, the mRNA expression of the gene does not vary that much from the wild type after at T0 generation. Then we check the seed phenotype of wild type and both our mutated lines. When we check the wild type residue seed under the light microscope, it looks clear and transparent and structure. But when we check our mutant seed, we found the seeds looks opaque and blur. So we assume that maybe due to the mutation, it looks like this. Then we want to check like what type of coding sequence is changing after the mutation. So this is the coding sequence of SB3 gene target of wild type, and this is the guide RNA region. After deletion of 1 base pair, we observed there is a frame shape mutation for overall coding sequences, and there are premature stop codon has been created. So we expected that due to this premature stop codon, non-functional protein were created, and we found deletion, substitution, and also insertion for all of our genes, target genes, though there might be premature codon were so our major findings here, we designed the guide RNA we successfully designed the guide RNA for all 4 genes, and it works both in vitro by the in vitro digestion and in vivo in our mature plants when we transform with Agrobacterium mediated transformation. And altogether, we achieve 100% transformation efficiency in tissue culture, and we found higher percentage of mutation, which is 78% after checking the mutation result. And overall, we found 10 plants where all 4 SBA genes, SBA genes are mutated, and we successfully used Amplicon sequencing through Miseq, which helps to analyze the result more efficiently. And finally, we observed the mutation effect of the mutation in seed phenotype. So there are some future goals for this project. We want to check the copy number of the transgene because it is very important, because we want to segregate it out the transgenes of the later generation for making transgene-free mutated lines. And then we want to check total starch content, amylose content, and resistant starch contents of the mutant lines, and also the wild type, how much percentage the amylose content is increasing, and check the yield parameter of the wild type. So if there is any negative effect after the mutation we got or something, and then an expression analysis of the targeted genes in both mRNA and the protein level of the mutant line. I like to acknowledge all the community members, especially Dr. Septi and Dr. Mike, for giving me the opportunity to work in this lab, and also Marco and Myra for helping us in tissue culture process, and all the members of our lab, because we have a big lab, everybody like cooperate if anything happen. So not last but not the least, our funding sources, Genome Editing Seed Grant, TAMU, and Texas A&M Agri-Life Research. Thanks to all. I'll be happy to take any questions right now.
Great presentation, Sudip. Very nice work. Questions? I see there's one in the chat. Devendra, why don't you ask it yourself?
100% is not right. Okay. Yeah. In case of like how much plant is regenerated, we just calculated based on this one, because you said 100% transformation and you mentioned is not right. Transformation and efficiency is calculated based on how much callus are transformed and how much transformed plant you generated. Okay, but I calculated based on how much plants are regenerated from the tissue culture, and if there is any like selection escape or not. But in our case, we found like all the plants are containing the transgene after the transformation process.
Other questions?
I have a question. Nice talk. I have a question about the white grain phenotype. Do you think that's coming from the change in starch species?
Yeah, like there is a one, through the literature search, I found that one of the group work on like starch branching enzyme 3 genes. They found the same type of mutation after knock out these genes. So we are thinking like due to the amylose and amylopectin ratio changing, starch structure is changing here. So maybe that's why it is happening. Like we are found more blur in structure.
Okay. So the reason I ask that is that going to negatively affect the commercial value?
If like it's easily like processing something, like if it make good after cooking, it give good food or something, or good amylose content or something, I think it does not affect that much. Like in that way, I'm thinking this.
Okay, thank you.
How many generations does it take to sort out the selectable marker?
What? Can you mention again?
You talked about segregating out the selectable marker. How many generations does it take to make sure you got it all? And
No, like
you get fragments of it when you sequence, you don't go enough generations?
No, like for the segregating out, first I want to check like how many copies of the transgene is there. I'm not yet checking yet. So I want to check now how many transgene is present there. So I want to choose only where only single copy is present. Then like with the Mendelian law, I will grow the plants and select the plants where there is no transgenes and only the mutation present. So in that way, I will select that plants only. So in T1 generation, I can select which plant does not contain the transgenes and which plant contains the mutation.
But that's based on the activity, the transgene.
Yeah.
What about getting rid of all the fragments, because they still make it a GMO? If there are any parts of that gene that are still in there, it's still a GMO.
Yeah, that's why like I want to check like the first generation to get rid of like any of the transgene is present in the T1 generation. So I want to check only the plants where there is no transgene is present, but only the mutation is present there.
Okay, thank you.
Sudip, this is Eugene again. I'm a soil scientist, so cannot understand your technology very well. But I'm very interested in your CRISPR technology. I just wondered, since you still use a virus as your vector, right?
Mm-hmm.
In your work.
Agrobacterium, yeah.
Yes, okay. I'm wondering if there are any other abiological methods to do those kind of gene editing instead of use virus or bacteria in your gene transfer here. Do people use some other methods to follow CRISPR and this technology? Do people use other vectors instead of virus?
Because like it is already well established, like Agrobacterium normally people use in tissue culture process. So this Agrobacterium does not affect anything in the like in rice or because it is not pathogenic bacteria. So, so although it is, we are using like control strain, which does not create any like pathogenic effect or something in the plants.
Oh, so basically your method can be used in any crops or plants, huh? Without any modification.
Yeah.
Thank you.
Welcome.
Other questions?
I had a quick question. Again, I'm not a gene person either, but from a starch chemist person, when you start using these, you were talking about, when you're transforming the ratios of the amylose to the amylopectin, that's probably gonna have some commercial issues from a processing standpoint, because the higher the amylose, the higher the gelatinization temperature. And sometimes you can get to a point where you even need pressure to cause gelatinization of that starch. So I was just wondering how if you thought, how is it going to impact somebody who's going to actually be utilizing the starch? Because if you want them to eat it, they're going to have to.
No, because we have lots of plants, like already like 30 plants and different combination also. So with the gene effect, we can also characterize like which gene give like minimum starch contents, like not that much effect, drawback after after components. So I'm thinking like maybe there are some beneficial effect also, like compared to, because we targeted 4 genes. So every genes have specific effect. Like, so we need to first check like which combination of the gene make best quality product, like with the starch contents. Then we can like make it go to the commercial way.
Great, thank you.
All right. I think we're out of time for questions. That was an excellent presentation, Sudip. It was very